Hi, I'm Stan Roux. I am Professor Emeritus at the University of Texas at Austin in the Department of Molecular Biosciences. I am partially retired in the sense that I no longer teach, which I did for 45 years at the University of Texas, but I am still active in research. I still have a lab team and we're still making discoveries and still enjoying it very much. I'm currently now interviewing at the Useful Wild Plants of Texas headquarters to talk about plants and my career loving plants and doing research on plants. My initial interest in plants really started in college when I decided to plant a tomato garden for my fraternity. And I planted 15 plants of a variety of tomato called Big Boy. And at that time I was living in Lafayette, Louisiana, which has very rich soil and a lot of rain and a lot of sunshine. And these plants grew amazingly. I started them growing March 15th that year, and they kept growing all the way through December 10th. And by the time they finished, they were over 8 feet tall, and I had to get a ladder to harvest the tomatoes off the top of the plant. And each of those plants averaged about 10-pound yield. So I gathered 150 pounds of tomatoes that season. And my fraternity brothers really enjoyed it, that's for sure. But the process of taking a plant from a seed to a giant plant that is loaded with fruit just amazed me. And I decided that although I was in college already and I was a classics major at that time, that I was going to, when I continued my studies in college, I was going to include biology because I wanted to learn more about how this miracle happened to go from a seed to this giant plant. And so I had a double major in college. Part of my college was in Lafayette, but then the final 3 years of college was in Mobile, Alabama at Spring Hill College, and there I had the double major of classics and biology. I actually also included philosophy as another major. So I graduated with 3 majors from Spring Hill College. And you know, I did well in my undergraduate, graduated summa cum laude, and I was encouraged if I wanted to go on and do graduate work in biology. So I decided to go ahead and do that, and my initial graduate work was at Loyola University in New Orleans, and I got a master's degree doing electron microscopy of plants. Really fascinating, enjoyed that tremendously. And as I was finishing up my master's degree, the chairman of the department at Loyola said, you know, I have really good contact at Yale University, a guy that I graduated with when I got my PhD at Vanderbilt. He went on to universities, and I went to Loyola. He went on, and he's now at Yale University where he's a professor. And I think if I write a letter for you, might be able to get you into Yale. So he did write the letter, and I did get into Yale. I had a pretty good record, so I also had a National Science Foundation fellowship, and that sort of paid my way through Yale. My program there was in developmental biology, so that really helped me fulfill my curiosity about how plants develop from a seed to a giant plant with fruit on it. And one of the things I learned during that graduate PhD work at Yale was the similarity between plants and animals and how they develop. The rules are very much the same. And that fascinated me, sort of the unity of life, that it's what you learn about plants in most cases applies also to animals. And that's really kind of fascinating, and it was a surprise to me, but I was delighted, because then I felt that if I went on in a career in plant biology, anything I learned would have applications, and not just to plants, but to animals as well. I could make discoveries that would be fundamental enough to be of general interest. So I decided to stay on at Yale for my postdoc work, which I went from developmental biology to biophysics, and learned a lot about membrane biology. And part of that was because the protein that I studied for my PhD was a membrane protein, so I wanted to learn more about how proteins interact with membranes. And at the end of my postdoc, I began looking for jobs, and in an academic setting, and I think I applied to 31 different places, and I got a couple of interviews, and one of them was at the University of Pittsburgh, and the interview went well, and I got the job at the University of Pittsburgh. And one of the interesting things there was that the lab I inherited at the University of Pittsburgh was a lab that the professor with whom I started at Yale had when he was at the University of Pittsburgh. So it's like, what a coincidence that, he and I ended up in actually the same space, doing research in the same space. So I was at the University of Pittsburgh for 5 years, and there's a lot of serendipity that happens in life. Everybody experiences serendipity, but it turned out that when I came up for tenure at the University of Pittsburgh, that coincided with the dissolution of my department that I had joined at the University of Pittsburgh. And I was now in a new department with a new chairman that didn't know anything about me, and was an animal biologist that didn't have very much appreciation for plants. So I said, uh-oh, this is not a good situation to be coming up for tenure. So I began applying to see if there was any other place that I might have as a backup in case the University of Pittsburgh didn't offer me tenure. And again, serendipity happened. One of the places I applied was the University of Texas at Austin. And it just so happened that the chairman of the department at the University of Texas at Austin at that time was a professor named Ted Delavorius, with whom I had had lunch almost every day when I was at Yale. He was a professor at Yale, and he and I got to know each other very well, but then he had left Yale to go to the University of Texas at Austin. And so I had a decision to make when the University of Texas offered me the job, because at the same time the University of Pittsburgh offered me tenure. And I had to think about that, because the University of Texas said, we will not give you tenure until you've been here a year or so and we can get to know you and know that you'll be okay. And so how many people give up tenure to go somewhere else? But I made the decision to give up the tenure offer at the University of Pittsburgh and go to University of Texas at Austin for two reasons. One was the University of Texas was a botany department, and everybody in that department appreciated plants. At the University of Pittsburgh, I was the only plant biologist on a faculty of 40, and none of the others appreciated plants, and I was constantly being asked, why do you study plants? And they didn't take very well the answer that I love plants and I wanted to learn more about them. That didn't seem to jive with them. So I knew if I went to the University of University of Texas, which at that time was ranked #1 in the country in botany departments, my work would be more appreciated by my colleagues. So that was one reason. But I have to admit, a second reason was they gave me a 33% increase in salaries. So I said, okay, that's a good reason. So I took the risk, and another reason why I took the risk, because Ted De Laborious, whom I knew was chairman of the department at the University of Texas, said, Stan, if you don't get tenure here next year, I will resign as chairman of the department because I believe in you. I said, okay, good enough. The risk seemed to be worth it, and it paid off. I did get tenure, and I remained at the University of Texas for the next 40 years, teaching and doing research, and I felt very privileged. So that's the long story. So some of you listening to what I've talked about in my career may be wondering, why did my colleagues at the University of Pittsburgh not appreciate plants? And the answer is maybe not too surprising. In order to do research anywhere today, you have to have a lot of grant money. And it's a lot easier to get grant money if you're working on AIDS or COVID or cancer or bacterial infections, anything that has to do with health. In fact, money awarded by the National Institutes of Health, which is largely focused on human health, is a lot more abundant than money offered by the National Science Foundation, which is more focused on basic research. National Science Foundation routinely funds plant research because they understand the fundamental importance of plant research for general biology and even for humans. But NIH is a little bit more directly focused on human health. And so they tend not to award grants to plant biologists. There are some, but I would say probably less than 5% of the grants offered by NIH go to plant research, whereas in NSF, in biology, probably 25-30% of the grant money goes to plant research. So there's that fundamental difference in attitude that says research is important only if it applies directly to human health. And it's not that important if it applies more broadly to basic understandings of life. And so my colleagues at the University of Pittsburgh knew this, and they knew that I was a pretty good scientist, and they knew that if I applied for NIH money, I could get it, no problem, if I was working on animals. But I was not likely to get it if I was working on plants. And so they didn't understand, why would I sacrifice grant money to work on plants when I could get a lot more and have bigger labs, more fame, perhaps, if I worked on animals. And you can imagine then why I was attracted to go to a botany department at the University of Texas, where I didn't have to explain why I was working on plants. But an interesting thing happened, which is at the University of Texas, which is happening, unfortunately, all over the country. And that is, as administrators of universities become more aware of the need for what is called overhead money that comes from research, they become more leaning toward hiring scientists that can bring in more overhead. That's how they pay the bills. That's how they keep the lights on. That's how they pay salaries. So it's not like administrators of universities have anything against plants. They may even have a deep love and appreciation of the value of plants, but they got to pay the bills. And so what happened at the University of Texas was that the plant biology department was dissolved and was merged in with, split into two other departments, one of which I joined was molecular biology, and the other one was integrative biology. And in both molecular biology department and the integrative biology department, the plant biologists were in the minority and had less say about the future of those departments. So you can imagine when both molecular biology department and the integrative biology department had opportunities to hire people, there was less likelihood that they would hire plant biologists than a botany department. A botany department would always hire plant biologists, but those departments had no allegiance to plant biology, and so they would go with whomever they thought was the best scientist, or maybe even whomever they thought would bring in the most money. So the reality, at least in the United States, I'm not sure about Europe or Japan or China, but at least in the United States, there is such a focus on bringing in grant money that the likelihood of plant biology departments continuing to be plant biology departments or of other departments hiring plant biologists goes way down. And so what is happening, if you compare the strength of plant biology in the university setting in 2022 compared to when I started at the University of Pittsburgh, 1973, there's a lot less strength in the plant biology field. And I think that's to the detriment of the health of biology science in the university, in the United States. I don't think, if I had to guess, based on my reading of the literature, it appears as though China has not bought into this attitude. They are certainly under the gun to feed a couple of billion people. And so they have a deep appreciation for plant biology in the terms of its agricultural significance. And they are pouring huge amounts of money into plant biology research. And so when you read the literature today, 2022, compared to when I started in academia, 1973, what you will see is that in the top journals now, 50 to 60% of the authors are from China. In 1973, it was 5% or less. And what this reflects is this change in authorship and in quality publications reflects the difference in funding in plant research in China versus in the United States. From a worldwide perspective, you can say, well, basic science discoveries are basic science discoveries, and who cares whether it comes from China or the United States? And I agree with that. And I deeply appreciate the great science that's being done in plant biology research in China. But somehow or another, I strongly believe, that it is the obligation of plant biologists in Europe and in the United States to convince the administrators of universities and the grant agencies of the critical importance of plant biology for humanity. And if, in order to do that, they need to demonstrate in some tangible way the monetary value of plant research, then okay, so be it. And I'm not trying to say we should all be just focused on the money, but what I am saying is that sometimes, in order to demonstrate the value of research, you have to relate it to some monetary term. Or at least some term that can readily be appreciated by the taxpayer, because ultimately they're the ones who are going to pick up the bill. So this is the challenge that I'm throwing out in a way to the scientists in the United States and in Europe who want to see plant biology thrive. And what I have noticed is that both in my own career and in the career of the plant biologist in my department, molecular biology at the University of Texas, there has been an increased amount of attention paid to convincing the granting agencies and the university of the value of plant research for humanity. As a consequence, amazingly enough, even though NIH rarely gives plant biology grants, 4 of the scientists, molecular biology scientists in my department have NIH grants. Why? Because they were aware that NIH wants them to convince them, NIH, that of the value of their research for humanity, for humans, and they did it, and they got money. So, when I started in '73, there was absolutely zero need to convince anybody about the material value or the humanity value of plants. You just did it because you wanted to discover more about plants. But now that's not the case. And I think once scientists recognize this, plant scientists, they can be very successful because you can easily convince objective, open-minded people about the value of plants. It's not a huge challenge. I mean, it is really valuable. And there's another aspect to plant research that can be emphasized maybe perhaps a little bit more, and that is because of the similarity of plant biology and animal biology, basic discoveries made in molecular biology and plants often have value in animal systems. And if it's okay, I would like to give a little bit of an illustration of that in my own career. So I've been funded by National Science Foundation. I was funded continuously for almost 40 years to do basic research in plant biology, never thinking that anything I did would ever have any practical value, because I just wanted to understand how plants grow and develop. When I was teaching a course in research to undergraduates at the University of Texas, I wanted them to have the opportunity to make fundamental new discoveries in plant biology, and so I had them do research on a topic that I was very familiar with, which was my own field. And in my own field, I was studying an enzyme. The name of it is apyrase, but you can call it anything you want. The enzyme apyrase, which we found could help cotton fibers grow better, and could help plants grow better, and could generally roots grow better. And so I was having the students do research on this topic, and one of the students in the course was very successful, and so he got an opportunity to come with a fellowship or with a stipend to do research as a freshman in his freshman year summer, after his freshman year of the university. But during that summer of research, he learned that he wasn't really cut out to do basic research in science, but he thought he would be better cut out in the business world. So he switched from biology to be a business major. In his junior year as a business major, he was given the challenge of designing a project that could make money. Business school, of course. And he remembered that this enzyme called apyrases helped plants grow better, and in particular helped cotton grow better. And he's a Texan, and he said, hey, you know, cotton's a pretty valuable crop in Texas. And I happen to have contacts with some people who have a little bit of money, and I think I can convince them to invest in this research to see if we can get better cotton growing in Texas. So he proposed that as a project, not only proposed it in business school, but actually made the contacts with these business people, and they agreed. Wow. So I got, from these business people, funding to do more research on how apyrase helped cotton fibers grow better. But at the end of the first year of funding, all of us, the business people, and I went to a cotton conference in Dallas. And at the end of that meeting, a scientist from Monsanto got up and said, I hate to throw cold water on what you guys are doing, because you're all doing cotton research, but it turns out that if you want to make real money in agriculture, you should invest in a crop that brings in a lot more money. For example, corn or soybean. So the business people who went to this meeting, who were funding my research, immediately contacted me after the meeting and said, hey Stan, why don't you see whether apyrase helps cotton, not only helps cotton grow better, but helps soybean and corn grow better. I said, sure, why not? So I did, and long story short, it does. And I've been funded continuously by this entrepreneurial group in Austin for 9 years, because the results from field trials indicated that indeed you can get more yield from soybeans and other plants by getting this apyrase enzyme to be expressed more in those plants. So let's take a step back from this experience. Funded by NSF for 40 years with no idea that anything I was doing had any practical value. But it turned out that when I discovered that apyrases make plants grow better, that had immediate application to the agricultural field, to the point where people who only are focused on money decided it was worthwhile to invest in that. Now that's just one story about how basic research led to a discovery that can help feed the world, can help overcome food insecurity in the world. And that story has been told over and over and over again in plant biology basic research, that discoveries made, for example, by Mendel in genetics, had huge implications for health for humans. And there are actual review articles that list all the discoveries made in plants that had tremendous applications to the health of humans. And so it's not like a news story or a surprising story, it's reality. And I think it's incumbent upon plant biologists to somehow convey this message to administrators, to granting agencies, that what they do does have semi-immediate value to humans, even though it's studying plants, even though it's studying fundamental aspects of plants. I didn't start off trying to find out how to make soybeans make more beans. No, I just wanted to know how plants grow and develop better, and I made a discovery that actually, unbeknownst to me or unsurprisingly to me, did have application in agriculture. So the take-home message here is that it should be easy to convince administrators, convince the tax-paying public, to convince granting agencies of the value of plant biology, not just for plants, and maybe not even just for agriculture, but for biology in general. And I think once that message gets communicated better, then I think perhaps there'll be a resurgence in strength of plant biology, not only as it is now in China, but as it could be in the United States and Europe.
Okay. I brought something up to you at lunch the other day. It'll give you an opportunity to talk a little bit about useful oil plants. And that was the little thing that we do when we do field trips for the classes that you had, and we would look at a lot of plants and that had certain qualities, and your students would, when I'd ask them what they noticed, they said, well, everything is little. You'd have to work all day to get a meal, something like that, which is a lead-in to the chili patine and all the peppers that Lynn had there. But if you could talk about the course and what we did, talking about the project, and then we take them on a field trip and show them all these different plants, and then stop on the chili patine. But it would also give you a chance to say, let's just say you wanted to isolate and enlarge on the production of any one of the things we looked at that was tiny. It would give you a chance to talk a little bit about some of the steps you'd have to go through to do that, and the cost to do it. Like this morning we were talking about costs, you need $100,000. Why do you need $100,000? What do you do with $100,000? So it seems like it might be a good segue to talk about that, and then maybe somewhere after that, talk about why you continue to support what we're doing for all these years. But that could be a separate talk, because this seems like it could end in explaining how you would get from a plant with a certain quality to creating a crop, and that'd be enough to, and the start of it could be how you had your class come out here and whatnot.
Mhm.
So I'm gonna stop talking.
So let me think a little bit about okay. So clearly it was very exciting for me to learn that my basic research did have some value in agriculture. And I tried, and I realized that this is something that because of my personal experience, I could more easily communicate to students. Not only in research, like in a laboratory, like the student who founded the company did, but also more broadly. And so I conceived the idea of teaching a course called Hidden Treasures of Plants, in which the theme would be to communicate the myriad different ways that plants have value for humans. And the course was an immediate success. The students, it was really funny, the students often started to get into my course not because they were interested in plants, but because it fit their schedule. And my course was offered on Tuesday and Thursday afternoons, and there were a lot of people in engineering, that was the only time they had free. So it was required that they take a course like Hidden Treasures, or there were like 100 different options that students could pick from. That was required. It was a broad-based course that the university required all students to take, no matter what. So this is a new initiative, was a new initiative on the part of the University of Texas, where they felt it was critical for every student coming into the university to have a broader base than just their narrow field. And so they offered 100 different courses that students could choose from, and one of them was Hidden Treasures of Plants. And for the first 2 or 3 years I taught the course, 60 to 70% of the students signed up for it, admitted that they had no interest in plants and were only taking the course because it fit their schedule. However, it turned out by the end of the course, every student was absolutely mesmerized by what they learned about the value of plants. And a highlight of the course for them every year was to take them on a field trip to the headquarters of the Useful Wild Plants of Texas, where there is about a 1 acre or 2 acre plot of land next to the office that had quote unquote weeds growing in it. And they would come for an hour from the University of Texas to the headquarters. They would get an introductory lecture from Scooter, and then they would go out into the field and be stunned by how the different quote unquote weeds in that field had real value for humans. And every year at the end of the course, students would say this was the highlight of the course, because it brought home the main message of the course, which was the hidden value treasures of plants. And when you go out into a field loaded with quote unquote weeds, there's no obvious value to those plants. The value that's there is hidden and can only be discovered by scientists who learn what the value of those plants are and try to promulgate that to others. And this is a key reason why I have joined the effort of the Useful Wild Plants to generate the volumes in the Useful Wild Plant Encyclopedia series, which does this job of promulgating the value of plants extremely well. And that value is illustrated many different ways in the encyclopedia. And so someone, and I use that as a textbook actually for the course, so that students wanting to learn more about how this particular plant or that particular plant that grows wild in Texas has value, they could find that value in the encyclopedia. And so when students were at the end of the course asked to write an essay on a discovery they made about the value of a plant that they chose, and I gave them that option of choosing any plant they wanted, they often found treasures of information in the encyclopedia. And so they came away from the course with a deep appreciation for the value of the encyclopedia series and of the efforts of Scooter and Lynn in the Use of Wild Plants to promulgate the value of plants for current generations and future generations. Now, I often wondered, how what I did in my research could connect somehow with some of the things we learned in the Useful Wild Plants Encyclopedia. And one of the stories that fascinates the students when we go on the field trip is we find a plant, a weed in the field called chili patine. It grows wild in Texas. And the fruit is a very tiny little red ball that's hot as hell. And they are told and are amazed to find that is the ancestor, as it were, of all the peppers that we now have today. The bell peppers, the various hot peppers that we have, all started out in evolution as this mother plant, the chili patine. So I often wondered, how would you learn more about the steps by which this tiny little pepper became all these various different kinds of peppers? And molecular biology would really help you figure that out. I haven't done that research yet, but maybe one day I will. But what you would do is you would look at the genes that are expressed in the chili patine compared to the genes that are expressed in, say, a bell pepper, and look to see which genes are in common. So that would be like the essential pepper gene, and then what genes different from each other that could have changed the size of the fruit and could have changed the heat of the fruit. And with today's tools, it would be very possible to be able to specify these following 10 or 15 genes played a major role in expanding the size of the fruit. And these 10 or 15 genes played a major role in decreasing the heat of the pepper. And so then you would have a kind of a map to predict if you wanted to select peppers, new kinds of peppers with different properties. You would know what to look for genetically. And you could either achieve those new peppers by crosses, which is the sort of traditional way of doing it, and using as your map which genes you want to emphasize, or you could do it by genetic manipulation. So those are the options that you would have, and those would be options you would have today. Now, if you were to pursue that research, or any research basically in plant biology or animal biology, it's very expensive. And to give you an idea of what I mean by expense, a typical small lab in any fields of biology would have a budget in excess of $250,000 a year. And why would they need that? So one postdoc, which would be a very small lab. There are many labs that have 4 or 5 postdoctoral researchers in their lab. But one postdoc, the total cost in overhead, salary, fringe benefit would be $90,000 just for that one person. And if you wanted to support a graduate student, one graduate student, which would be a small lab, one graduate student, one postdoc, another $40,000 right there. And then how much does it cost to buy supplies just for a lab? Typical small lab, not a big lab. $3,000 a month. It's another $36,000. So it's expensive. But you know, when I think about what society spends money on today, and you look at the budget of NSF or even the budget of NIH, maybe a couple of $2 or $3 billion a year for both of them combined, that's a drop in the bucket compared to the $1 trillion budget that the United States spends on tax money, or more than $1 trillion. So we're not talking about a major percentage-wise percent of investment of tax money, but boy, the return would be amazing.
One thing to say that's a little different is when we do that field trip, we look at 80 to 100 plants in 30 minutes. And each of those plants has some unique qualities. And one approach is to take the chili patina and compare it to the peppers that have been developed or selected from it over thousands of years. But the other question that might be more important to us is, can you justify looking at the favorable qualities of these other species and enhancing those through molecular biology? And because that opens it up to 400,000 species, whereas if you just look at the chili patina peppers, it's a very contained thing. It has to do with our current commercial thing, but if you think of it on a world scale, it's galactic. I mean,
yeah.
So it might be important to say as a part B, having talked about the chili patina and all the other peppers that we use,
apply that principle to all the other plants, right? Yeah. Okay. So the story I told you about chili patina and comparing it to the peppers that we have today, all the other peppers, that same story would apply to any of the 400,000 different plants we have on Earth, where you could say, wow, here's a plant that has amazing drought resistance. Why? Why is it drought resistant? Because, man, I tell you what, especially right now in Texas and in other parts, in the western part of the United States, California, being able to grow when you're under drought stress would be an incredibly valuable quality. So how would you learn what is that drought resistant gene that allows plants to do that? So when you go into the field, like we do on a field trip, and we see maybe 80 or 90 different plants. Some of them are still green, even if, we're in a drought. And that happens. We've been doing this for many years, going on these field trips, and sometimes there's not much rain, but we still see green plants. Why? We could look at that quality and say, okay, here's plant X that has this quality of survival in drought. What is that quality genetically, and how can we discover it and apply it to crops that we want to be drought resistant? How could we do that? Molecular biology would give you the tools to do that. And actually, as an aside, it turns out unexpectedly that this apyrase gene that we study not only helps soybeans produce more seeds, but it also makes them more drought resistant, which is a totally different quality that you wouldn't have expected. And yet, if you make that discovery, you can begin to make the connections. Why would this apyrase gene give you drought resistance? Well, it turns out it makes the roots grow better, and it helps them take up water better. And so those are indirect qualities that are maybe not directly related to drought resistance, but are critical for drought resistance. So that same process of looking at a field of plants and looking for qualities in that field that you want to have in a crop plant, for example, or in any other plant, there are molecular tools that would allow you to discover the basis of that quality and then to apply that to plants that you may be more interested in.